So it's obvious that selection plays a central role in maintaining biological species. It acts to eliminate those alleles that cause hybrid unfitness, that cause individuals to be unable to survive or to find mates. But on that view, speciation seems difficult. If we think of species as living under Cyril Wright's metaphor on different adaptive peaks, then how is it that they can get from one state across a valley of low fitness to another state, to another adaptive peak? And that apparent difficulty in seeing how species can cross from one peak to another has stimulated all kinds of theories which rely on some kind of random process, some kind of random genetic drift, to knock a species into a new state despite the opposing force of selection. But in fact, the difficulty is more apparent than real, that selection need not oppose divergence if the reproductive isolation is not expressed during divergence. And indeed, naively, one would imagine that diverging populations would naturally become incompatible after a sufficiently large time. One way to think about this is to see that as separate populations accumulate genetic differences for whatever reason, they become eventually able to produce a whole variety of new genotypes which have never been produced before, which have never been tested by natural selection. And so if we think of these two lineages accumulating different substitutions, A, B, C in one lineage, X, Y, Z in the other, if there are n different substitutions, let's say 10 or 100 distinguishing them, only a similar number of genotypes are ever produced during their divergence, of the order of 10 or 100 distinct genotypes during their divergence. And yet when they come together to produce an F1, the F1 has never been seen before, and the F2 and the backcross genotypes have again almost all never been produced before, never been tested by selection. And so we naturally expect hybrids to be less fit, simply because they involve new, randomly combined genotypes that have not been tested against selection. And this idea that reproductive isolation will occur as a natural byproduct of divergence was in fact first suggested by William Bateson in 1909, but is now known primarily as the Dobzhansky-Muller model, after Dobzhansky and Muller, who in the 1930s, 1940s put forward this idea. And it's been developed in great detail since, particularly by Alan Orr and Michael Torelli, who've estimated that actually there must be a very small probability that any particular pair of substitutions interact to cause trouble, to reduce hybrid fitness. And indeed, what's surprising about speciation is that it's so slow, that populations that differ by hundreds or even thousands of amino acid substitutions can still produce viable and fertile hybrids. Speciation occurs over a timescale of the order of millions of years, typically, whereas over that time, there are very, very large numbers of differences which potentially could cause trouble. So what's surprising is the robustness of organisms to genetic change. And, of course, we can ask what causes divergence, what causes, in a sense, speciation, but that's really a special case of the general question of what causes evolution in general. How much, say, amino acid sequence change is due to selection rather than drift or whatever. But given that a substantial amount of divergence is driven by selection, it's then easy to see that divergence can occur rather readily in parapatry, despite gene flow. In a sufficiently broadly distributed population, even quite weak selection coefficients can maintain locally adapted alleles. And so if we extend this idea of inevitable allopatric divergence to a parapatric system spread over a rather large area, then virtually all of the mechanisms that work in allopatry will also work in parapatry, provided that they're driven by moderately strong selection. Okay, so this is a sort of very straightforward, really, view of the evolution of biological species, the evolution of reproductive isolation. But in a way, it's rather boring, because reproductive isolation by itself will simply fragment a species into a mosaic of small local subspecies or species which cannot coexist with each other, because when they do, the rarer type will suffer more through hybridization with the opposite type. Reproductive isolation by itself tends to produce a mosaic of parapatrically distributed species, which ultimately become smaller and smaller and smaller and eventually go extinct. So by itself, reproductive isolation really seems a bad thing. What we need is along with reproductive isolation, the evolution of sympatry, the evolution of the ability to coexist, which requires the ability to use different limiting resources to fill different ecological niches. And that's what I'll concentrate on in this talk. So to be sympatric, we have to have both reproductive isolation, so that different combinations of alleles aren't reshuffled all the time by recombination, but we also have to have the ability to exploit efficiently different limiting resources. So I'll set up a very simple model, really a very simple extension of the classical Levene model, in which we have a number of niches, a number of different microhabitats, say, and we have really three components to the model. I'll really just think about two niches to keep things straightforward, but we can obviously extend it to more. We have, firstly, habitat preference. We have a probability that any particular genotype will choose to lay its eggs, let's say, or move to or exploit this resource versus that resource. So these probabilities, alpha 0, alpha 1, add up to 1. They're the probability of going to here or there. And then once the individuals arrive at a particular microhabitat, they have some net probability of surviving in that microhabitat, V0 and V1. And these are just probabilities of surviving from, let's say, egg to adulthood. And then we assume strictly soft selection. We assume that the output of each niche is fixed by the amount of resource. Okay, so this is a simplification, but a reasonably good place to start. And so we say that the proportions coming out of each niche are fixed, C0, C1, independent of the composition, the genetic composition of the individuals living there. And in principle, we could allow for mating across all niches. We could have adults emerging and then milling about at random. But I'll assume a situation more favourable for speciation, in which mating occurs on the microhabitat. So we might imagine, say, the classic Rhagolitis example, the true fruit flies, in which larvae develop within apple or hawthorn or cherry or whatever, and then emerge as adults and mate on that host. So there's mating within the microhabitat. So that there we have habitat preference and differential survival as magic traits which inevitably lead to reproductive isolation, because mating patterns are associated with host use. And the very simple model I'll use is to translate this model of habitat preference and viability into a quantitative genetic model in which additive traits can determine those two features. And because habitat preference and viability are limited to a range of 0 to 1, and yet we want to use machinery of quantitative genetics with Gaussian distributions on an infinite range, we have a transformation in which we use a very simple function. Let's see if I can work this. Yeah. In which the probability of going to niche 1 rather than niche 0 is given by this logistic function. That will be essentially 0 if y is negative, will increase in a sigmoid function towards 1 if y is positive. So y ranges from a very low value to a very high value, and as it does so, we have this transition from choosing to go to one niche or the other niche. And in a similar way, we have viability given by a similar function, where z now is a continuous variable inherited, let's say, as a result of a number of additive loci. And we again have this sigmoid relationship between viability and this underlying trait z. But we also need to include some kind of constraint, some kind of trade-off on performance on one niche versus the other niche. For habitat preference, there's a very natural trade-off because you have to be somewhere. This plus this must add up to 1. So there's a linear trade-off for habitat preference between choosing to go there or choosing to go there. So if this plotted the probability of going to niche 1, probability of going to niche 0, they have to add up to 1. But for viability, that isn't the case. We can have, in principle, any combination of viabilities, but we expect that there'll be some outer limit, some constraint on what is possible. And we might have an outwardly curving constraint there, or a downwardly curving constraint there. And the shape of this trade-off curve is determined by this parameter beta. So if beta is less than 1, then we have a situation where we can have a generalist. We can have an individual that does rather well in both niches. Whereas if beta is if beta is greater than 1, then we have this situation of specialization, where there's a strong trade-off. Either you do well there, or you do well there. Being halfway in between is worse on average than being a specialist. Okay, and that makes a crucial difference to the outcome. This is all really the classical Levine model. Okay, so mostly I'll be thinking about a single population, but at the end I'll say something about the parapatric case, in which we have potentially a decline in resource availability. The sizes of the niches may, at any particular point, be roughly equal, or they may vary across a linear gradient, with mostly this niche here, that niche there. And then we could add in some sort of local dispersal and look at the interaction between gene flow and selection in relation to niche use. Okay, so what's going to happen? Well, habitat preference has to evolve to match the sizes of the niches. If individuals are all choosing to go to one particular niche, then they'll be relatively crowded, they'll compete more, they'll do worse, their fitness will be lower. And there's very, very strong selection to balance out the exploitation of the niches against the productivity of those niches. So we expect that habitat preference will evolve to strictly match the habitat size. The average preference, the average of this parameter alpha over all of the genotypes must equal the output of that niche. But there's no constraint on the variation in preference, and so we'd expect actually the distribution of habitat preference to be possibly highly variable. The distribution may be very broad. There's no selection on it, except that the mean has to be matching the output. That's not the case for viability when beta is not equal to 1. If we had a linear trade-off for viability, then actually the population genetics are identical to the population genetics of habitat preference. But in general, we'll have either this situation or this situation, and the outcome depends on the sign of that curvature. If we have an outwardly curving constraint there, then typically there'll be a single generalist genotype evolving, which exploits both. But more interestingly, if we have this negative curvature, we'll have selection favoring, ideally, a bimodal distribution in which we have a mixture of genotypes specializing for this or that. Okay? So, and the relative abundance of these two specialist genotypes will match the output of those niches. So that's what we expect if we forget about genetics, if we assume that anything is possible, if we have essentially asexual reproduction, so that all possible phenotypes can be maintained. But recombination will prevent the evolution of specialist genotypes. That selection will be acting in a disruptive way here, but recombination will be pulling the population together to produce a rather narrow distribution. And we'd expect that if we think in terms of this underlying additive trait for either viability, then at each underlying locus, we may maintain polymorphism for alternative alleles at equal frequency. Okay, so we'll have strong balancing selection. But if we have balancing selection on a number of loci, then with free recombination, they'll not be able to generate the extreme types that would allow specialization to here or here. So provided that the maximum variance that can be maintained by selection on all these different loci, but with linkage equilibrium, with random association, provided that that is not sufficient to allow specialization here, then the population will, if you like, be frustrated. It'll be sitting in this sort of generalist space when it would actually be much fitter to specialize in a bimodal distribution, to speciate. So we have to have some means by which we either get some sort of bimodality, either by reducing recombination or by some kind of cunning genetic trickery. And so we can get this bimodality occurring in two ways. One would be by the evolution of modifiers that increase the strength of preference, that reduce the rate of recombination. In the extreme case, we might even have a shift to asexual reproduction, which would allow the efficient exploitation, the matching of genotypes to the appropriate available resources. But if that isn't possible, then we'll also have selection for coupling between preference and viability, and also for coupling between different components of viability, between these different polymorphisms, which are maintained by balancing selection. So the picture we have is that we may have a population which is under balancing selection, which maintains large numbers of polymorphisms. But if that population is freely recombining, so that those different polymorphisms are being reshuffled all the time, then we may not see any great divergence. We may see a more or less homogeneous looking population. And yet, if somehow those could get coupled together into two alternative combinations, matching alternative niches, then we will see essentially speciation. So how do we get to that point? Okay, so this distinction was in fact first raised by Joe Felsenstein in a very influential paper, A Homage to Santa Rosalia, in which he termed these two alternatives a one allele versus two allele scheme. One allele here because a single modifier allele can produce bimodality in some way, by reducing recombination, increasing preference, or whatever. Whereas when we have a two allele system, in other words, where we have to associate different alleles with different niches, then recombination is opposing the process. It's much harder to get speciation. So, on this second view, in which we have this so-called two allele scheme, in which we have to get a coupling, a linkage disequilibrium between alternative components, we see that we want to couple these polymorphisms. And indeed, that can happen if there's sufficient genetic variation for traits that influence viability or preference. And in some ways, this has been seen as an example of reinforcement. It's sometimes been termed reinforcement. I think there's a bit of confusion about the semantics here, or at least disagreement. But in fact, it's quite different. This process by which an initially homogeneous population, if you like, condenses out into two distinct types through the increase of linkage disequilibrium. Although it was referred to in Felsenstein's paper as a kind of reinforcement, it's actually rather different. And the key difference is that selection can act at any stage. We can have the coupling between different factors, all of which involve selection against survival or against fertility, that is post-zygotic factors. Whereas in the classical view of polymorphism, of reinforcement, in which we think of a modifier increasing associative mating or reducing recombination or whatever, we think of pre-zygotic isolation being strengthened in order to reduce the loss, the load, from cross-mating. We think of an allele, let's say, which increases assortment because it prevents the loss of offspring through mating with the wrong type. And so we have the classical distinction between pre-zygotic and post-zygotic isolation, which applies only to this sort of one allele version. It doesn't apply here, where we can talk about the coupling together of any kinds of incompatibility, both pre-zygotic and post-zygotic. We can imagine a population as containing genetic variation, which may be, if you like, cryptic, hidden, because of the process of recombination. And the strengthening of linkage disequilibrium, the emergence of two distinct species, is, maybe it's a kind of reinforcement, but is not reinforcement in the classical way. Okay, so what actually happens in this model? Basically, the outcome is very simple. We can model the genetics using the so-called infinitesimal model, in which we say that two parents produce offspring that on average are halfway between the parental values and that have a distribution that has a fixed variance, which is essentially generated by recombination, by the segregation of different polymorphisms, assuming large numbers of alleles. And in fact, this works very well here. If we model the explicit genetics with 10, 20, 30 loci, you get almost the same result as if you assume this very naive, almost phenotypic model, in which we just say that parents produce offspring with a distribution of trait values. And the key parameter here is what we call the variance at linkage equilibrium. And that goes into the model by saying that the variance between individuals within a family is half of the total VLE. And if we had no selection, we just let the infinitesimal model run, then the within-family variance and the between-family variance are equal to each other, equal to half VLE. So we have this one parameter which tells us the strength of genetic variation for ability to adapt to or to exploit the different niches. So this gives us a fairly general way to think about the problem. If we have a linear trade-off, beta equals 1 here, then we can measure the, make this a bit bigger. We can measure the strength of reproductive isolation that ensues by working out the probability that an individual that survives, that emerges from this niche, was born in a different niche. So we take a random individual and we say, did it come from a different place? Okay, was it in a different niche in the previous generation? And that varies smoothly with the standard deviation of underlying variance in trait use. And the scale here is set by this logistic curve that determines the viability, ranging from 0 to 1. It's a fairly smooth transition if we have a linear trade-off, but if we have strong disruptive selection, in other words, this downwardly curving trade-off between viability here and viability there, then we have a rather sharp threshold. This is for beta equals 2, beta equals 4. We have a sharp threshold value of this variance. Below that, we have a unimodal distribution. Above that, a bimodal distribution. I should say, though, that this sharp threshold behavior, which allows strong reproductive isolation, depends on a crucial assumption. And that is the assumption that a specialist on one niche does very badly on the other niche. So if you're perfectly adapted to one, the original model, the simple model, said you died on the other. Okay, so there's a complete trade-off. If you change it by adding this theta parameter, by saying that, well, if you're perfectly adapted to this one niche, let's say if z is very, very large and positive, you still have a probability theta of surviving on the other niche. Okay? So specialists can manage, even if rather badly, on the other niche. And it turns out that even if this theta is quite small, say 0.2, it almost eliminates the divergence and eliminates the reproductive isolation. You get rather weak isolation. So quite small levels of survival on the wrong niche can greatly weaken the disruptive selection that drives the process of speciation. So just to look in detail at what happens, suppose we choose some sort of trade-off beta equals 4, which is a fairly sharp trade-off between one niche and the other, and we increase the standard deviation of this underlying trait that determines how well adapted you are to one or other niche, from 0.57 up to 0.63, a fairly small range. Just below the threshold, if you start with a high variance, you collapse down to a low variance. This is the variance of the underlying trait value over time. And what that means is that you start with a bimodal distribution and you collapse down to this unimodal distribution. These curves here show the survival of this niche or that niche as a function of trait value. So this is the underlying trait value, which is determining whether you survive well here or survive well there. If you're above the threshold, then if you start with a low variance here, you expand out and eventually, after a few hundred generations, get to a bimodal distribution. So you've had speciation. In between, there's a rather narrow range where you have two alternative stable equilibria, where if you start up with low variance, the unimodal distribution, you stay there. If you start with a bimodal distribution, you stay there. But it's actually rather hard to get that alternative state. That may be somewhat model dependent, but it seems, at least in this model framework, typically there's a rather narrow range. If you're below that variance, you don't speciate. If you're above it, you do speciate. So we have a rather simple model in which we have a single threshold. If you have enough genetic variance, you'll speciate. But if you take that model and then ask what will happen in parapatry, what will happen if you have a gradient in availability of the alternative niches, then it becomes much easier to get a shift. The threshold variance goes down. So one way of looking at this is to think of one deme here that has one niche, one deme here that has another niche, and a deme in the middle that has both niches at equal frequency. And then individuals from here and here will migrate in. They'll generate excess variance. The mixing of divergent populations will generate excess variance, will assist the process. So even if there's a rather small underlying variance contributed by the genetic system under the infinitesimal model, linkage disequilibrium generated by migration will inflate that variance and will trigger the process of divergence to exploit different niches. So with this 2-deme scheme, we have a situation here. We again plot against the standard deviation of the underlying trait at linkage equilibrium, the reproductive isolation up there. This is what happens with beta equals 2 with no migration. And if we increase migration even quite small amount, 0.02, 0.1, 0.5, we end up with a rather sharp threshold with migration at its maximum value of 50% migrating. We end up with this rather sharp threshold at a much lower value, less than half as much, about half as much, as in the absence of migration. And you can do the same thing in a continuous framework. Let's suppose we have a continuous gradient in resource availability, which has a certain width. As we make that width narrower, and we're making the width here narrower, going in this direction, the strength of reproductive isolation increases, and the threshold genetic variance required for a transition also decreases. So again, speciation is encouraged quite substantially by divergence variation in niche availability from place to place, which is a fairly obvious point. Okay, so I think it's amazing, I've almost kept to time. Okay, so I'll try and summarize here. I'm sure Isabel's talks are going to be much more interesting, so I don't want to eat into her time. Okay, I said that selection inevitably is involved in speciation. It is responsible for eliminating hybrids, for the sexual selection that causes mate preferences that separate biological species. But that kind of selection need not be expressed during divergence. And it may be that, although selection is involved, it drives divergence for quite different reasons, quite unrelated in general to the speciation that eventually occurs as a byproduct. But selection has to be directly involved in the evolution of sympatry, which in some ways is arguably the more important, more interesting process of speciation, the process that generates real biodiversity, real diversity within one region. And here we have to understand the tension between disruptive selection and recombination. And the key process is a coupling together of different components of reproductive isolation. And I've really just summarized a very simple model framework in which we can see that there's basically, in this way of looking at it, a single parameter, the underlying variance in niche use, in adaptation to different niches, which determines the threshold at which you can get divergence. And I've said finally that this process is much more likely to happen in parapatry than in sympatry for given parameter values. So, of course, there are lots of open questions here. It would be very interesting to extend this to more realistic genetics, to allow, for example, the output from each niche not to be fixed, but to depend on some kind of explicit density regulation. But I think it's an interesting way to start thinking in a more systematic way about processes of sympatric and parapatric speciation. Thank you.
Thank you very much, Nick, for a very interesting talk and also for keeping so well to the time schedule. So you are entitled to a full question and answer session, and we have wireless microphones. So if you could please raise your hand if you have a question, and then we will hand you a microphone.
Yeah.
Well, we also have to switch them on, these wireless microphones. Not yet. You have to press this mute button for long.
Shout.
Yes.
Yeah, people always work in models, as you do as here as well, where you have to assume change in recombination. However, people never assume pleiotropy. It would also be possible to say that you change some aspect of the perceptual system. You would change both viability and mate recognition. But it's never done in these models. Do you know why?
No, laziness, or people haven't got round to it. I mean, there are all sorts of interesting extensions like that. Although, in this framework, actually, in the population genetics, what matters is the product of preference times viability, alpha times V. So, in a sense, you can think of all these different components of fitness on this niche or fitness on that niche, which of course are biologically significant, biologically different, but think of them as in one combined quantitative trait. And so in some ways, this way of looking at it sort of combines these different components that you're thinking of. But if you're thinking about the actual process of mate choice or host use or whatever, then you do need to think about the distinct components of the things that I've lumped together here, really, as preference viability.
Yeah, the thing is, one has to think in terms of what these animals really can do.
Right.
For some reason or other, people dream up easy mechanisms.
Yeah.
Because I have the feeling that if one would dream up other mechanisms, it might well make it easier, for instance, where you have linkage of the mechanisms in one gene.
Right. So you could certainly easily extend this model to have a trait that determines preference for different hosts and a trait that determines viability. But I mean, it's hard to see that those would be determined by actually the same gene in most cases. I mean, you then start to think about recombination modifiers and so on, but you're thinking of something that really has pleiotropic effects on distinct components.
Effects. I think they are rampant, actually, at least when one tries to translate what the evil people say to what ecologists say, there's very little match. That's the reason why I ask this question. I think this might be brought in.
Yeah. I think it's really it's an empirical question. We need to find what the genes are and find how far they are pleiotropic.
Let's see whether we have any other people who want to ask questions. Any hands? Perhaps I could add a quick question about the evolution of habitat preference. It seems to me that this is perhaps a rather easily evolvable trait. So many morphological traits, many physiological traits might also have side effects in terms of habitat preference. So once we allow habitat preference to evolve, could you comment on how it would affect the conclusions?
So, you can imagine all sorts of things affecting whether you end up here or there, affecting both the strength of preference and the direction of preference. But there is a problem in that if you prefer to go to a niche where you just don't survive, then of course it's selected against. So you do need this coupling between the two. And so I'm assuming in all this framework there is abundant variation, but even if there's quite high levels of variation, you still need a coupling in order to get the process started. That's a fundamental problem.
It would seem that evolution would provide this coupling quite naturally, right? So with the co-evolution of the local adaptation affecting viability and the habitat choice affecting where you end up during dispersal, we would expect this coupling or this matching habitat preference, so you would get it as a result of evolution instead of having to assume it, right?
Yes, and I mean, one point is that it would be very much easier to see that co-evolution happening if there's variation in habitat availability. So in a certain area, there's a lot of this host, right? And then, of course, selection will be acting to bring together all of these different things. And that's another distinct reason, I think, why parapactory is absolutely favourable, at least some variation in habitat use.
Okay, there's one more question over here. Maybe we can try the microphone again. Make sure it's switched on. It does project very well.
I just wanted to make a comment on the last conversation that you two had, which is important, and you raised, as usual, many interesting questions. But this one about the co-evolution of the habitat preference may not be so difficult as once we thought. I personally believe, and there's much behavioral evidence, that organisms have mechanisms to measure how well they're doing in different environments. Those are general. And when you change phenotype, you will still have that same process, and it will feed back to you the information that you need to have to adjust where you go in the environment.
Yeah, that's a very good point. It might be interesting from that point of view to try and do a comparative study of groups where you're committed to a particular host, where the egg is laid there, you can't do anything about it, that's where you are, versus where you have a choice. And then you can be intelligent, you can choose to go somewhere tasty.
Okay, wonderful. Thank you very much again, Nick.